Hi guys, today I'm going to help you make your own cheat sheet for Mendelian and non-Mendelian genetics. On your agenda, you can click on the cheat sheet, open it up, and here I've got the backbone for a cheat sheet for you. You don't need to use this, you can make your own, you can do it digitally, make a copy, or you can do it by hand. Personally, I'd prefer to do it by hand. That way you get a little more flexibility in what you'd like to put on here. But we are going to go over the difference between homozygous and heterozygous. So homozygous being same and heterozygous being different. Recessive and dominant, we should all remember from general bio, the dominant letter being the large letter, the recessive being the small. Homozygous would be two of the same, and heterozygous would be one large and one small. So you can have homozygous dominant, where they're both large letters, or homozygous recessive, where they're both small letters. Those letters are going to represent our genotype, the root of this being gene. And as genes are microscopic, we use letters to represent them. That's compared to phenotype, which is what it looks like. P for physical characteristics. So a genotype for a tall plant might be big T if tall is dominant, little t for recessive, and the phenotype would be tall or short. An allele is a different form of a gene. Each of those alleles would be a letter. We all know the Punnett square, where we would put our letters on the outside. So if I was crossing two heterozygous tall plants, I'd have a big T and a little t crossed by a big T and a little t. It doesn't matter, but we always put the large letter first so we don't get confused thinking that big T, little t, and little t, big T are different when they are in fact the same. Pedigrees, if you guys remember from general bio, we have the squares representing the males and the circles representing the females. If it is completely colored in, it means they have the disorder or the trait. If it's not, it could mean that they're not, or they could be a carrier. Some pedigrees show this as half shaded in, and some you have to figure out from the pedigree whether or not they are a carrier, kind of like this pedigree here. So we have non-Mendelian genetics, where we have a couple of different terms. Incomplete dominance means it's not completely dominant. For example, if I cross a red flower and a white flower, that heterozygous combination would be a pink flower. Codominance, they're both dominant. For example, blood type. If you have an allele for type A blood and an allele for type B blood, that child would have codominance and they would have AB blood. Multiple alleles means there's more than two. Polygenic traits and sex-linked traits we'll get to a little bit later on. Your sex-linked, we tend to see this more with males. It's almost always on the X, and that's why we see it more with males. With females, they have 2X, and males, they have XY. When you have two of the X's, you're more likely to be a carrier to pass on to males. The dihybrid crosses get a little more complicated than the monohybrid that we saw above. And I have an example here that I think explains this pretty well. And we will work with this more in a separate video if it is in fact confusing. So here, this would be a cross between two heterozygotes. And you'll see that foiling here, which we'll talk about in a later video. There's space at the bottom if you would like to put any other things on your cheat sheet to use as a reference. And as we go along with the multiple videos we'll have for tutorials and our practice problems. If you go back to your agenda, you will see an example of my cheat sheet that I have filled out for you. So kind of what we've explained in this video, just in print. So you can double check yours against mine or use mine for help if you would like. So you can see that foiling that we'll go over in a future video.